All right, everybody, we're back for part 2 of our Spectronaut tutorial. So just to recap, on our first video, part 1, please refer to it if you want to. We went over looking at the tree view, the grid view, how to reorganize the tree view based on strip sequence. We redid it with protein group ID. We looked over the grid view and looked over how to look at your data. So today we are going to go over how to change some of the fundamental settings in Spectronaut, like how to change from 1 peptide minimum to 2, maybe turn on and off imputation, some fundamental things you could change on your end. And then we'll go over the post-analysis view. I have about 40 minutes today, so we're going to have to stop at 40 minutes because we have our Proteomics Clubhouse at 11:30 and it's 10:40 on a Thursday. So for those of you who don't know, check Twitter. Ben Neely, Nist, and I have a nice every other week Clubhouse chat for proteomics. So check that out if you want. We talk about all sorts of cool things and stupid things. Okay. So let's go over how to change some of the fundamental things processing, how a Spectronaut does its processing. So I am using the viewer just like you are. So I am not using anything proprietary with the commercial non-viewer software. So the viewer can do almost 95% of what we do with the non-viewer software, but there are a few things that it limits you to. So what we're going to do is, remember, we talked about the secret right-click menu. So we're going to make sure we're on analysis. And again, I am going a little fast, but remember, you can always pause and rewind and fast forward. So because it's streaming, feel free to do that anytime. So I am going to use my secret right-click mouse button and click over, make sure you're on the analysis tab. I think it only works here, and right click over your Spectronaut file tab here. And you'll see stuff like remove runs, map missing runs, add runs. You can see this is grayed out, so that's something you can't do in the viewer, I think, but I can do it if you want to add runs. But if we find outliers, and I'll show you how to find outliers, we can remove the runs later. You could refresh the post-analysis, order runs by group, all sorts of stuff. But first, you can see here where raw files are missing. So overnight, I left this open, and I think my external drive disconnected. So we are going to remap the raw files. So we're going to go map raw files, click that, and you can see all of these ones are missing. We'll go browse, and I believe I put them here. Data, let's see, where did I put this? I think I put them under Spectronaut, and here they are here. You hit OK, it finds them. The red little checkbox, little X's turn green, yay. Then you hit OK. And now it stops complaining. So again, we usually, it's 50-50 that we return these data files to people. So for example, just yesterday, someone was asking me about Spectronaut and the data files, and there, I think it was like 5 or 6 runs or 7, and it was like 30 gigabytes. So sometimes we don't do it, but like we say, we're really happy for you to have them. So if you want them, please ask, and we will give them to you, and we'll be happy to give them to you. Okay, so let's look at some of the fundamental settings we could change in Spectronaut. So we're going to go to the tab, secret right-click. We're going to go to settings. All right. So there's some things that are grayed out that you can't change in the viewer, I believe. So these are all grayed out. So let's look at the things we can change in the viewer. So identifications we can change. And the things we can change is exclude single hit proteins. So the rule of thumb is, for some people we check this box and some people we don't. It kind of depends on the day and how we feel, and like kind of looking at your experiment and what you're doing. We're like, well, maybe we should do this, maybe we shouldn't. So usually we do, but sometimes we don't. It really depends on the group. So you'll want to definitely check exclude single hit proteins if you want. And what this does is it limits your proteins to a minimum of 2 peptides per protein. Okay, so what would you want to do? Why would you want to do this? Well, there's definitely a trade-off between sensitivity and quantitative accuracy. So if you uncheck this box, you will identify more proteins in your sample. So probably it will increase between 20 and 30%. So the bad part about it is sometimes those are what called one-hit wonders. They're more likely to be wrong, but they're usually pretty good. We're filtering on a 1% false discovery rate, so they're usually good. But the problem is you may not get a high quantitative accuracy out of these single hit proteins, but sometimes they can be really important. So on the safe side, check it if you're looking at quantitative profile differences between like a lysate or something between lots of different proteins. But you may want to uncheck it and go back and look for important proteins, because I have seen some projects, just one the other week, where the protein they were looking for was just identified by a single peptide, and it was super important. And that was the protein they were looking for. And if you check this box, you would totally miss it. So definitely play around with it. Uncheck it, check it, look at your data. If you're missing proteins that are super important in your project, then definitely uncheck it, see if it's there. But if you're looking for proteins that are changing globally, like a global profiling experiment, probably safer to check it. Okay, but we're going to leave it unchecked for now. Or maybe we'll check it. Okay, let's check it. This you can leave p-value estimator. We just kind of leave that at default. These are the FASTA files you can use. It looks like you can import another FASTA file. I've never tried that before. Let's see, gene annotation. You can add Go gene annotation, gene ontology information. I usually don't change that. Condition setup, this is important. So if you want to change your conditions, you can do that here. So you can see we have, there are several different boxes. We have one called reference. So you can make those your control if you want. And Spectronaut is even smart enough to, if your sample names have control in them, to, I believe it will automatically select the control for you. You could change the run label if you want. So this is just a label it puts on some of the figures, maybe the reports. I like to leave it the way it is because it's less likely to get lost. But for example, if you wanted to change, you just double click. And if you wanted to change this to like 25 minutes, because these are the 25-minute runs, or 45 minutes, or whatever, you can name it condition 1, condition 2, or whatever you want to do it. These are the conditions. Oh, and if you change the run label name, it will still be linked to the file name here, so it won't get scrambled. But it is a little bit more cumbersome to go back and find what's what. Okay, you could change the conditions here. You can see we have 2 25 minutes, 2 45s, 2 60s, and 2 90s. And remember, this is the same 50 nanograms Promega K562 that we use as a QC, and so these are just run at different gradient lengths. Okay, and this is the replicate. I believe this sets it for you, or you set it, but it'll automatically set it for you. Quantity correction. We never really change this, but if you know, for example, in one condition you've loaded 1.2 times the amount of protein, you can put that there. Usually we normalize before, so we try to put the same amount, but you can play around with it if you want. I've never really had a reason to change it. Label, you can change, I think, yeah, 45 minutes. So these are the labels on some of the figures, so you could change those. You could set the color, so you right click on this, and then you can click on this and select whatever colors you want. It's pretty nice. Can't change the file names because that's set by the file name. You can export the condition setup. So let's say if you have 8 runs, this is not cumbersome to do by hand, but if you have like 100 or 200, what you'll want to do is export the condition setup. Change this to, it'll save as like a TSV file, even though it'll end in like an XLS. Why do people do that? I don't know why people do that. Anyway, so it'll save as a TSV file. You can load it into Excel, edit it as a template, and then re-import it into Spectronaut. So it's very convenient. So that you can change. So let's see. Summary. Here's the summary. So let's go back to analysis settings. So we looked at checking the exclude single hit peptides. We're going to keep that check. We're going to check it. This we usually don't change. This is single hit definition. What this really means is that it'll count a single hit if it's modified. So like, for example, if you detect a peptide with an oxidized methionine and one without, but it's still the same underlying sequence, that means it's only one hit. But if you change this to by modified sequence, then I believe it'll count it as two if there's a modified and unmodified. All right, quantitations, what we want to look at. These are all interesting things you could change. Usually we don't change them, but you can. You could change them if you want. Usually the protein LFQ method, the label-free quantitation method, within the last version, Spectronaut 15, I believe it changed to LFQ. LFQ is a well-established label-free quantitation method technique that the paper came out. I think was from Max Quant. I have to read this paper like every other month to remember it, but it does a really good job, and so it usually does that by default. Okay, let's see here. You could change all sorts of things. We usually leave them default. One of the things we do change is changing the data filtering. So by default, Q-value sparse and global imputing are turned on. So this is good for if you're doing t-tests or you're doing some sort of statistical tests that don't behave very well if you have missing values. So like, for example, we run into this problem all the time because a lot of people here do viral infection experiments or protein induction experiments where one group of samples should have, like if you're turning a protein on or off in one condition, and you have one condition that should have this protein expressed and the other condition it doesn't. What this setting, the default setting, will do is if it's missing in one condition, it'll do an imputation method, which is basically, some people say it's like making up data, which it's really not. It's just kind of like determining the background and doing some sort of fancy imputation. You kind of want to watch out. Some imputation strategies are not very good for proteomics because it'll affect the coefficient of variation. But this one is pretty good. We turn it on by default, but if you're a biologist and you're doing an experiment where you don't expect proteins in a certain condition, it will really like confuse you because you'll be like, wait, why is my viral proteins in the control? Did I screw up the experiment or my technician's doing something weird? I'm like, no, it's just because global imputing is turned on. So definitely turn that off if you want, and sometimes we will turn it off if you tell us the specifics about your experiment and we know it should be turned off. But for the global profiling and for normal tests like a t-test, usually it's better to turn it on, but there's some controversy to that. So you could turn it off by changing this to Q value. So this kind of gives you a tooltip of what they all mean. So Q value, changing it from Q value sparse to Q value, you see will automatically turn off imputation. So what this does is it means the peptide has to be identified in the run in order to quantify it. Q value sparse means it only has to be identified, what does it say? At least once. So all it has to do is be found at least once in any of your experiments, and then it will do an imputation of the background if it's not found in others. So you will always get a signal for every single protein in every single sample. So if you do a Venn diagram of your different conditions, they will all be overlapping because every protein will be in every sample because it will be imputing. So you could turn that off. So we'll turn this off. Cross-run normalization, we usually leave this on by default. But if you have normally, so normalization is an important thing to think about. So this will normalize all samples so that the overall signal is the same. So this is good when you expect most of your proteins, like 90% of your proteins, not to change, which is normally what you do for global proteomics profiling. But there's some experiments where this will really screw you over. Like, for example, an IP or something where, let's say, you cause apoptosis to happen, and half your proteins in one set of conditions aren't there anymore because they're all dead. So like all your cells are dead. So normally we leave this on because global proteomics profiling really works better when only a small majority of the proteins are changing. But there are certain proteomics experiments where this isn't true, like IPs or something where you expect global proteins to change. So another example of this is like viral infection experiments. So at the beginning of the virus infection, you don't expect the cells to proteins to change radically. Maybe you expect a small portion of them to change, but later in the infection, maybe the cells start dying, right? So the global protein is going to change. So you want to watch out for normalization and Q-value and data filtering for viral infection experiments, other sort of infection experiments, or experiments where you expect global proteins to change. So we're going to leave cross-run normalization on, but feel free to turn it off and try it. So we're going to turn off imputation. We're going to put it from Q-value to Q-value sparse, Q-value sparse to Q-value. You can also play around with Q-value complete. This will really decrease the amount of data in your experiment because I think it requires the protein to be found in every single sample for it to be quantified. Maybe you want to check that out. So Q-value percentage, you can do that too, and you could say, hey, if it's found in 20% of the samples, do it. But we're going to do Q-value. Okay, let's see. So we're going to change that. So PTM workflow, you could turn on PTM localization. This is only really useful if you're looking for a PTM like phosphorylation, ubiquitination. Those kind of things work great for Spectronaut does a great job for those with this kind of DIA data. So, but for if you're doing global profiling and you just have like the normal oxidized methionine turned on, you don't normally want to do it. Okay, so protein inference. We usually leave this as is. You can see you can do inference algorithm, ID picker, or all matching proteins. We leave it at ID picker. Probably the method for this is in the ID picker paper, which is, I believe, is David Tabb's paper, I think from a long time ago. Basically what this does is this tells you, because remember we're dealing with peptides, this tells you how to go back to the protein level from the peptides. And it's not an easy thing to do. So, for example, like I think I mentioned this in the last video, if you have peptides that are in more than one protein, for example, why we have protein groups, and this is kind of tells you what algorithm it uses, which is actually very important. Okay, so post-analysis, what can we change here? Calculate, explain, tick. I don't really know what this does. Let's see. Extensive, we'll use expensive. Is that expensive? We'll use an expensive. Okay. Feature detection method. Well, cool. Why wouldn't you want to use an expensive feature detection method to calculate the most accurate result for the explained tick? Cool. No wonder it's so expensive. Okay, let's do that then. All right, let's try it. Calculate sample correlation matrix. I'm going to check this. It's kind of cool. Let's see. We will just leave this as is. Assume equal variance. That's for your t-tests. It defaults to unpaired t-test or paired. And I think this is why they like imputation, because I think for t-tests it works a little better. Gene ontology, run clustering, z-score transformation. You could do a z-score transformation. Okay, so let's go back and look at what we changed. We changed exclude single-hit peptides. We changed this from Q-value sparse to Q-value. What else did we change? Did we say anything else? Oh, we turned on calculate explain tick because we want to use the expensive feature detection because I paid for the software. Might as well use the expensive one. I think that might be a typo, but I don't know. Maybe it is expensive. I don't know. Okay, I don't actually know what this does. I've never done it before, so let's give it a shot. And we turned on calculate sample correlation matrix because that's kind of cool. It does take some time, so I think that's why it's off by default, but let's see what it does. All right, then we're going to hit confirm. And it's going to do some, if you look at the bottom here, it's going to go through some recalculate a lot of its stuff. So let's see. And my hypothesis is that data that looks like this will disappear because I believe this might be imputed, and good data will remain. So let's see if I'm right about that. I'm not exactly sure. AES, I don't even remember where this plot is anymore. Let's see. All right, so this only takes a couple minutes. Hey, it's done already. Cool. All right, so let's see if some of these data, some of them still don't look very good. Let's see, but this, I don't know, I guess so. Sometimes you have to just, the computer does a better job than your eye, depending on the algorithm. You know, some of these your eye will go, well, of course, this looks great. And some of these, you'll be like, what, really? Like, is that a good result? I don't know. It doesn't look very good. Maybe you want to exclude this from your, let's say you look at this peptide and you're like, that doesn't look very good. I believe, what does this mean? This means something. I believe this means something. I don't know what it means. So let's see. Let's click on this. Here we go. I don't know. This might be real. So look, this is one example that we saw yesterday where it doesn't look good in this run, but it does look good in some others. So maybe you want to keep it because you don't want to exclude this peptide because it does look better in some experiments than others. Like it definitely looks better in the 60-minute run than than this one. There's something wrong with this 45-minute run anyway. Oh, we're on the 25-minute run. So I think we're looking at this one. But if you look at maybe this one looks better. So anyway, be careful about excluding it or not excluding it. So let's look at, this is actually interesting. Let's see. So this one has a Q value of 4.35 to 10 to the negative fifth. So that looks good, but let's see what it is here in this 25-minute run. Come on. Did it not like it? Okay, how about this one? Okay, so you can see the Q value is less, just over the limit, I think. You can make these more stringent, but it's borderline. You could see how the data is not very good, but it's, you know, borderlines here, so. Okay. And we can tighten up these Q values if you want. But some of them, these look good. Like, look at how nice this data looks for this one. And you can see how they vary across. You can see there's something wrong with this 45-minute run. All right, so let's see. We can go back, and I don't remember how many proteins we identified with the including single hits, but we can look at this if we click on the name of the file. And remember, we can go to, and this will I'll show you this in the next pane. Analysis summary. And so you can go up here and say we're now at, for this file, we're now at 4,926 protein groups, which is still really good for like 50 nanograms. 46,000 peptides. So you can kind of go back to the previous video and see how that changed. Okay. So we went over the grid view before. We showed you how to change this from use log to scale to not. You can see, for example, how some proteins look like they're differentially expressed based only on their retention, based only on the gradient. This is what some of these I expect. Some of them are interesting. Like this one, protein had a higher response in the shorter gradient than it did at the 60-minute gradient. Why is that? I don't know. That's a good question. I have some theories. This one, for example, these are definitely, this protein looks like it's more expressed in the 25 than in the 90, but I bet we'll find some that are different. And you could see how some of them are missing now. Do you see how we have NAs? So this is why we turned off. This is what happens when you turn off imputation. So you could see some are missing. You could see that 45-minute run didn't look good. And there's one missing from here too. So we'll start.
Starting your workout in workout.
What? What is it? Siri, I don't know what you're talking about. Okay, so let's see. This is interesting. Why would it do that? Okay, so this one looks like it's actually higher. You get a more response for this protein in the 25 than the 90. Why is that exactly? We could probably look at things that we can find the opposite. And remember, the 25-minute run did not have enough scans across the peak. And you can see how we get NAs now, huh? Where we didn't before. That's interesting. And you could see here is the opposite example where we have some proteins that are actually higher. You have a better response for them in the longer runs than you do in the shorter runs. And these may be because maybe you have more peptides. I don't know. It's interesting. Okay, and remember we can click on these and look at the data. It will now show you things that are not very good and things that are. Look at this one, look at the data. You can kind of get an idea of what looks better. Like, look at that. Isn't that beautiful? It's beautiful. So some things I think will get less crappy looking data because we're not doing imputation anymore, but still we see some that are borderline. Oh, and look, do you see how this is red? So this is showing you ones that do not pass the Q value, and you can see this doesn't. You see how this is red here, and you see how it kind of looks like crap, which it does. But remember, this may be found in a different sample with a higher Q value. So that's why it's not going to count it in the expression of this protein in this sample, but it will count it in one of another sample, which is probably what's going on. I wish you can right-click this. Or let's see, I wish you can click on these somehow and go to that other tree view and look at it across runs. That'd be nice. And I don't think you can. But maybe for the next Spectronaut, if you're watching this, that'd be really cool. So like one of the things I would like to do is say, hey, this looks kind of crappy in this sample, but how does it look in another sample? So right now I have to go back to the tree view, search for this peptide, or maybe there's an easier way to do it. Search for the protein at least. But it'd be nice if I could click on this and somehow, or maybe you can and I haven't. Maybe there's a secret function somewhere. All right, so we have 20 minutes left. I'm probably not going to make it through the whole video, but let's go briefly through the post-analysis view. Okay, post-analysis requires a refresh. Okay, so let's refresh it. All right, so if it says this, just give it some time. And you can go back to the analysis thing to see what it's doing. So once this gets done, and here's our ion mobility. Isn't that cool? That's cool. I like this. I like this graph. Good job, Spectronaut. Okay. All right, cool. So post-analysis. Let's look at some of the post-analysis runs. And I think I only have 15 minutes left, so I think I'm going to stop here. And then after our awesome clubhouse, I will be back to tell you about the post-analysis. And then maybe we'll do the report in the same one. All right. Okay, bye everybody. I'll see you. It'll be instantaneous for you, but it'll be like an hour for me. So we'll go over this in part 3. All right. I'll see you in like 30 minutes or 10 seconds or however long it takes you to click the next link. Okay. All right. Bye everybody.